So now we're going to talk a little bit about what is genetic engineering. And this is something that is really fascinating. I really enjoy talking about it, but can be extremely complicated. So I'm going to break it down a little bit for you guys and talk about the process of genetic engineering, how we use it for biotechnology. We're going to talk about how scientists copy the DNA of living organisms. We're going to explain how we can make recombinant DNA and how it's being used. And then finally, we're going to talk about transgenic organisms and how they are produced. So let's start off with how we got into genetic engineering. You know, when they started looking at microorganisms and plants and even other big organisms, they want to look at the DNA, which is really important, and see if we can find any mutations that lead to disease. But it's really hard to just sit there and look at your DNA and say, oh, this is a gene that leads to a disease. So the first problem we had to do was find a gene. And to find the gene, there are different ways we could do this. Once we found the gene, if you remember, central dogma is DNA makes RNA, which makes protein. Remember, protein defects are what causes diseases, basically. Well, once we found a gene, the DNA, we could then predict what the RNA was going to be and then figure out what would happen to the protein and how things didn't work. So again, how do we find a gene? Well, that's where these guys came into play. These are jellyfish. And if you notice, the jellyfish have a nice, pretty little color to them. Well, what's cool is that DNA from every organism is the same exact 4 letters. Hopefully you remember those 4 letters are A, T, C, and G. And we can put those in different combinations in different organisms. It's still going to read it as a gene to make DNA, to make RNA, to make protein. So scientists were able to do is cut a piece of the DNA from these jellyfish and put it into a bacteria or even into some other organisms. They've done it actually to cats, they've done it to mice, and they can follow genes because they light up in the organism and see what it affects in overall. So that's really what we're dealing with biotechnology. So what are some of the steps to get DNA? Have you ever watched those crime scene TV shows where they sat there and found a hair just sitting maybe at the scene and they're like, oh, I can get the DNA. And what they're really doing is they're taking the DNA from the root of that hair. But that's not enough DNA to do all the work that we need to do. We have to amplify it. We have to get lots and lots of copies of DNA. And we're going to do that using this technique called polymerase chain reaction, also known as PCR. And almost every single scientist I know doesn't say this name. They say PCR. So what happens? And we can do this in the lab. Actually, at the school, we have the techniques to do this. But what ends up happening is they take your DNA and they're going to heat it up. And the reason why they're going to heat it up is because when you heat up that DNA, it's actually going to separate the bases that are down the middle. Those are connected by hydrogen bonds. And we're going to get 2 pieces. So we're going to get this piece right here, and we're going to get this piece right here. Now, when you do a PCR experiment, you want to amplify or make more copies of one specific part. So they add a little bit of what they call primer and a whole bunch of A, T, C's, and G's. So if this is an A over here, we're going to add on onto this side the complementary letter, which is T. If this is a C over here, we're going to add a G over here. And if this is a G over here, we're going to add a C. And you kind of get what's going to happen. You're going to find the base pairs to add on, and we're going to use an enzyme. It's going to make another copy. So from one piece of DNA, after one copy, you end up getting 2 pieces of DNA. But the cool thing is we do it again. We heat it up again. So from the original, you make, so this one ends up separating out. We make from 2 copies, you end up making 4. And we do it again. And we do this about 40 cycles. And when you keep doing it, you're going to increase your numbers exponentially. So you don't just make like a linear, but you start off low and then you get really high numbers and you get tons of DNA in a short period of time. So that's one of the reasons why when you watch those crime scene shows or any type of analysis like that, you do see that you get lots of DNAs and they can do lots of experiments with those. But scientists are going to amplify your DNA, the DNA, and do some other things with it too. One thing that scientists can do is they can make what's called recombinant DNAs. And recombinant means, this is just a fancy way of saying recombine. If you think about this word recombinant, hopefully you can see the word recombine in it. That's all. We're recombining DNA. And we can do that from different sources. So we can take the DNA of a bacteria, mix it with actually a human DNA, and this is going to be a recombinant that can make us maybe a human protein that we can use for a drug. For example, insulin is done this way. We use the bacteria as these little factories to make human insulin protein that we then can isolate and be very, very pure, so there's no bacteria background at all. And then this is used to give to people who need insulin injections. So how do we do this? Well, we have a tool called a restriction enzyme. This is a special enzyme that will cut DNA at a specific sequence. So every time this restriction enzyme, this one in particular is echo R1, whenever echo R1 sees the sequence GAATTC in the DNA, it's going to cut it in a staggered way. So what you end up getting is what we call sticky ends. And the sticky ends, remember, these are going to hydrogen bond wherever it sees another, for this, if it sees another AATT, it wants to hydrogen bond with it. And this is another sticky end. So let's say this came from a bacteria, and this was the human insulin gene. You can cut this one with the same restriction enzyme. It gives you the sticky ends of AATT. This gives you the sticky ends of TTAA. When you mix them together, they're going to find each other, and we're going to add a little glue that we call DNA ligase. So again, I mentioned that we can do this with insulin to help make lots of insulin. We're going to use a special piece of DNA that we call a plasmid. This is found naturally in bacteria. And again, we can put that insulin in the middle of the plasmid, and kind of shown here, here's the bacteria cell with the plasmid. We isolate the plasmid, we cut it, we add our gene of interest. In this case, they're using the example of growth hormone. That's another drug that we can purchase that is grown this way. We cut off cut those up with the same restriction enzyme, mix them together. You get a new plasmid, and then we're going to put it into a bacteria, and then the bacteria is going to grow lots and lots and lots of copies, and all those copies are going to have that human growth hormone in it. Now, all that technology, like I said, DNA technology has kind of started. We've been doing this stuff since the 70s and 80s, but recently, in the last about 5 or 6 years, a new technology came out, and this is called CRISPR. And you're going to watch a little bit about this coming up too. But CRISPR is basically a way to edit DNA. So how do we go about doing this? All right, so here we have an enzyme called Cas9, and this enzyme doesn't have real scissors in it, but it has enzyme capabilities in there that can cut. That's why we show those little scissors there. And what's going to happen is we get a piece of target RNA sequence. So the scientists find out what they want to target. Let's say there's a gene that causes, and actually they have done this with sickle cell anemia. We know the sequence of sickle cell anemia, so they can make an RNA that corresponds to the part they want to fix. So the scientists take that piece of RNA, they mix it with this enzyme Cas9, shown here. They add it to the target DNA sequence. So let's say someone has sickle cell anemia. It's a mistake in the DNA. That we know exactly how to fix that mistake. We just have to change one base. Well, when you mix these two together, the Cas9 joins up with that DNA, it's going to cut using those little scissors we show, okay? And then it's going to make a nice little double cut, and we can do a couple things. Either we can disrupt the gene and kind of do a random repair to turn off the gene. This would actually turn it off. So if we have a gene that's making a protein that's bad and we don't want it to make any more of that bad protein, this can turn off genes, okay? Or we can add another new sequence with it and change it. So in the case of sickle cell anemia, this is what they did. They added a new piece of DNA, and they were able to edit and correct it so the person can function and not suffer from that disease anymore. So this has really changed a lot of how we're treating or looking at how to treat genes. Again, I know it has occurred with sickle cell anemia. I have not heard it to fix any other diseases as of right now, but don't be surprised if you start seeing this occurring a little bit more in the future. We also can use biotechnology to help change plants too. So here we have an example of what we call, it's a transgenic plant. So again, we start off with a plasmid, okay? We make a recombinant in there, and we add in a desired gene. So let's say we want to make a plant that makes extra vitamins. So for a country that might not have a lot of food, you can take a rice plant, add extra vitamin-producing genes into it, put it into the recombinant plasmid, pop it back into the cell, and now we grow these little individual plant colonies in a petri dish and eventually grow them to full length. And that would produce a plant that has the ability to make rice that has extra amount of vitamin A. So if you have a population that is lacking vitamin, you can give them rice and they can get all the nutrients that they want. I can also tell you we can do this with other organisms too. I did something very similar to this for fruit flies to create a different type of fruit fly. And if you've ever seen glowfish at the pet shop, that's exactly how they got these fish to grow. These are transgenic animals. They have been put in there either sea anemone DNA or jellyfish DNA to bring out these pretty colors that you see in them. This does not occur naturally. Now it's kind of cool to use these in the put in your fish tank, but there's actually a useful tool for a scientist. One thing that was done in the 90s was also cloning a mammalian animal. The first animal to be cloned was a sheep called Dolly. They called her Dolly because they were able to take the nucleus. I believe they took it from a mammary gland, and so they thought that Dolly would be an appropriate name for her. So what they did is they took an egg donor. So they took this egg donor and they removed the nucleus from this cell. Okay, so we have just a cell with cytoplasm, no nucleus. Then they took the cell donor, used the nucleus from that one, and fused them together. So this had the nucleus from this sheep, but it was put in the cytoplasm from this sheep. And there are two different species of sheep, so they do look very different. They did that on purpose because they wanted to make sure that the progeny, the baby, that the clone that came out would look like this. That was their prediction if they made a clone. Well, they fused the cell, fertilized it, made an embryo, placed it into a foster mother who was, again, a different species than the cell donor, and produced a clone lamb that was Dolly, and she was identical to the mom. Now, when I say identical, genetically identical, she didn't grow up to be exactly looking like mom, and one thing that they learned was that her DNA, so cell donor mom was 7 years old when they used her, and genetically, this little baby Dolly also was genetically older. She was about 7 years. She died of an early death because her DNA was older overall. So they learned that there is some things to think about when you're dealing with cloning. We can clone other animals. We have done a cat. That cat was called Copycat. Kind of funny. There's been a dog that has been cloned. The first dog that was cloned at the, I think it was Seoul National University, so they called it Snoopy, to kind of go with the Seoul National University. That's why they called it Snoopy. And we've been able to clone other animals too, but we do know that they're not exact copies of the mom and dad or the mom or whatever. And there are some things to think about and put into considerations when you do that cloning. So hopefully this gives you a brief overview of biotechnology, and we'll go into a little bit more with that for discussing ethics.